This is TWIS. This Week in Science, episode number 880, recorded on Wednesday, August 17th, 2022. Will we return to the moon? Who knows the answer to some of these questions? I'm Dr. Kiki, and today we will fill your head with sleep, sneezes, and styrofoam. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. Your mind is made up. It's made up of physical, biological, squishy stuff that came to be over millions of years of evolution, giving you the ability to process information from the world around you through your senses as it operates biological functions automatically. It's made up of thoughts, language, and memory, giving you the ability to wonder aloud and eventually find where you left your keys. And it's made up of interests, things your mind seeks out in the world beyond its squishy confines. While your mind is made up of many things, we recommend one more. Make your mind up with science. Like a highly refined sixth sense, science allows you to process information not readily available to senses alone. And there's no better place to make up your mind than here on This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. And apparently it is episode 888, even though I have decided we're going back in time again first. This episode.
Justin just said that's the kind of time travel you don't want to do.
No, that's the dangerous kind. Time travel forward, all you want. Going back's where all the paradoxy danger happens.
One step forward, two steps back. No, we're going to move right on into our science for the day. I have some fun stories today. I'm going to be talking a bit about cancer, synthetic. What?
I got fun stuff to talk about today. Well, let's start off with cancer.
Yep.
Yep.
That's what we're doing. Synthetic circuits, some styrofoam recycling, and is consciousness solved, and should we be thinking about free energy? Some big questions out there. Justin, what'd you bring?
I've got why heat makes you sleepy, especially if you're a fruit fly, ancient DNA of South America, finger tapping revulsion, and how deceptive food labeling could be countered.
Deceptive labeling with truth. No, let's hear that when we get to that story. Yeah, Blair.
Spoiler alert: Kiki just told how my story ends.
How about we just tell the truth? Blair, what's in the animal corner?
Oh, I have sponges, spiders, and zebra finches.
Oh, I love zebra finches.
Yeah.
Meep. Yep. I will be mimicking a zebra finch during that story.
It's actually zebra finch brains, so I brought it just for you. So you can basically do the story.
No, I couldn't, but I'm going to love it so much.
And you're going to, it's great.
I was
like, I got to bring this.
Oh, yeah. I didn't see that story, so I'm glad you did. Yes. Let's talk about those zebra finch brains later. But now we're going to move on into the show. And as we do, I want to remind everyone out there that if you are not yet subscribed, you can do that by looking for This Week in Science, all places that podcasts are found. You can find us on Twitch, on where else are we? YouTube and Facebook, broadcasting live weekly at 8 p.m. Pacific time on Wednesdays. Whether or not I get the episode number correct.
Mhm.
And then
Who
we
really
are,
cares
yeah,
about episode numbers?
it's just about the
Wait,
science, not the
What
numbers.
actual episode is it? We don't know.
I made
That's
it up a long time ago.
no, that's fantastic because
We've
that
talked
means
about this trivia before.
we've gotten to the point when we've lost track of where we are.
I mean,
It's
it's somewhere in the
real time.
800s
No,
or
900s.
900s.
It's an estimate.
Yeah.
And if you want to find us on the Twitch, the Instagram, or the Twitters, look for TWIS Science. Our website is twis.org. If this was a lot for your brain and you just want to go to our website and see our show notes and episodes and stuff and find links and information, twis.org. Okay, science time. Let's dive into the science. What do we want to talk about first? How about the fact that NASA has successfully rolled its Artemis 1 mission craft, the Orion orbiter and the SLS, onto the launch pad earlier than expected. It's sitting there on the launch pad, hopefully getting ready to launch on a test mission, not manned, near the moon, which is pretty exciting if they actually can get this test mission to go for it.
I don't know though. A man
what do
on
you
the
think?
moon.
Yeah.
A man on the moon, please.
Man,
Just-
well, so far
Just
it
couldn't possibly be done. Why are we?
couldn't possibly. No,
Haven't we already done this before? I feel like deja
we-
vu.
it has been done before, but not in a very long time. It's very exciting if we can successfully manage this unmanned mission to the moon and back, because then we could take people to the moon. We could take new rovers to the moon. We could do lots of stuff on the moon. We could start building that moon base that we've been talking about. There's all sorts of research that could be done. I mean, let's become our own little human I guess it's not interplanetary, interplanetary body.
Interorbitary?
Really?
Yes,
A
I think
United
they-
Federation of Planets, I think, is what you're looking for there.
Maybe that's what.
Yeah.
Yes.
Mhm.
We could get there. Yes, but no sooner than August 29th will the launch take place from Kennedy Space Center, Cape Canaveral, Florida. And so we're just waiting now to see whether or not everything goes according to plan. Will we make it to space? Will we return to the moon? It's definitely yet to be seen. But moving on from the moon, we can take a little shift in our scientific focus, to curing cancer in
Yeah,
mice.
let's do it.
In mice, because
Again?
as we know, this is what we love to do: cure the cancer in the mices. In this particular situation, they are testing a new kind of cancer vaccine. It is similar to others that are in clinical trials currently, as it works similarly to COVID vaccines that were used by Pfizer and Moderna by delivering mRNA in little lipid molecules to fuse with cells that then read that mRNA and produce viral antigens, these little tiny segments of the virus that then can make the immune system reactive. Okay, we know there are many of these mRNA vaccines for not just SARS-CoV-2, HIV, and cancer in development. Like I mentioned just a second ago, cancer vaccines using mRNA are in clinical trials. However, so far, what they have seen is a limited turnover of the actual immune reactivity when the mRNA gets processed mainly through the liver. Now, the liver is a great immune body organ in our body, but the better location are our glands, the glands that swell up when you get sick.
Oh yeah,
Yeah.
little sponges.
The
for
lymphatic system,
Yeah.
yes, and through the lymph glands, what we can what if we can target the mRNA to end up in those lymph glands, in the lymph system, that's where the immune cells are really doing the biggest work to prepare the body to fight something off, maybe we could have a bigger reaction. And so these researchers at Tufts School of Engineering, they attempted this, and in their publication and Proceedings of National Academy of Sciences this week, they showed that it was extremely successful. And rather than have, as with the COVID vaccine, 1 to 4, where like the majority of the mRNA is ending up in the liver, they had 3 to 1, where the majority was ending up in the lymphatic system. And not only that, the mice that they treated with this particular mRNA vaccine for their cancer, for metastatic melanoma, they showed inhibition of tumors, 40% rate of complete response, no tumors at all, and no recurrence long term when it was paired with another therapy that helps prevent cancer cells from suppressing an immune response. So all the mice were in that ended up in complete remission, prevented any new tumors from forming after they were injected again with metastatic tumor cells. So the researchers were like, let's try really hard to give these mice cancer. And they didn't get it. Nice. So this is a stronger response. It's a very exciting platform for potentially not just cancer treatments moving forward, but also other viruses and pathogens that we might want to deal with. Yeah,
that's awesome.
Yeah. So once again, in mice, but
in mice. Yeah, well, that's where we start.
In mice.
Yeah, exactly. But we'll get there. We'll get there eventually.
Cure the mice of all diseases, so that one day they may live forever, replacing us on this planet.
They will. The mice and the cockroaches. We will have saved them.
But this is just real quick. We've talked about this already probably enough times on the show about how we used to like try to avoid talking about cancer, not because we didn't want to talk about cancer, but because it was always a thing that might lead to a thing, to someday a pathway to finding an intervention, blah, blah, blah.
Yeah.
These are clinical trials that are curing cancer. And this is not the first one we've had this year alone. We've had, I think, several this year already.
Yeah.
Really an astounding time in cancer research.
It absolutely is.
You know what I was just thinking about too? Yeah. Pets. Cancer in pets is like a huge thing. Like, just
I guess as pets live longer, and as these animals we love, I don't really think about it.
Yeah, I mean, obviously humans are the number one priority. I get that, but I'm also just thinking about if we can cure cancer in mice. Maybe
what about zoo animals?
And our fuzzy buddies.
Yeah.
Well, yeah, and if your pet is a mouse, you're way ahead of the game.
Truth.
Press release on this next story caught my attention with the opening line. On the hottest summer days, you may find yourself dozing off in the middle of the day. That caught my eye because it's been especially warm in Denmark. It's 5:20 in the morning and I'm sweating. Like it's that, there's no air conditioning in this country. Like nobody has it. So it's been hot. And I did. In the middle of the day yesterday, I took a really long nap while it was really warm. And so I saw this headline. I was like, oh, well, why is that? So I took the long midday. Why was that? Northwestern University neurobiologists have found clues as to what might be happening. Their study, published in Current Biology, found that fruit flies are pre-programmed to take a nap in the middle of the day. Follow up to a 2020 biology paper that identified a brain thermometer, which was only active in cold weather. This paper explores a similar thermometer circuit for hot temperatures. This is quoting Marco Gallio, who's associate professor of neurobiology, and I think it's his lab that this is being conducted in. Changes in temperature have a strong effect on behavior of both humans and animals, and offers animals a cue that is time to adapt to the changing seasons. The effect of temperature on sleep can be quite extreme, with some animals deciding to sleep off an entire season. Think of a hibernating bear. But the specific brain circuits that mediate the interaction between temperature and sleep remain largely unmapped. So they're working with fruit flies because they said it's a good model. There's for the question of like, why do we sleep, or why does sleep, what does sleep do for the brain? Because they don't attempt to disrupt their own sleep instincts as do humans. No fruit flies drinking coffee to pull the odd all-nighter. They also allow researchers to isolate the influence of external cues like light and temperature on specific cellular pathways. So nice friendly model to work with. Just as they expected, based on the results of the previous paper on cold temperature, researchers found that brain neurons receiving information about heat are part of the broader system that regulates sleep. When the hot circuit, which runs parallel to the cold circuit, is active, the target cells that promote midday sleep stay on longer. This results in an increase of midday sleep that keeps flies away from the hottest part of the day. So, if you run one of the cultures that have that midday siesta, where you close up all the shops and everybody's just going to go take a nap.
I know. I was just going to ask, is this like, is this a genetic circuit in Italians as well?
Well, yeah. I mean, it might be generational, like growing up in warmer temperatures. You just sort of figure this out. But there must be a biological. But that graphic you have up there is showing that the fruit fly likes a good 25 degrees C, or was 77-ish Fahrenheit. And then if it's hotter than that, it triggers that response that starts saying, okay, well, these cells are going to stay on until you take a nap. The question
was that a guess, Justin? Because I just Googled it and that was exactly correct. 77.
He lives in Denmark now.
Yeah, I'm in the Celsius. Everything's in the Celsius.
Right? Okay, got it.
Yeah, I'm on the metric temperature system, which is actually completely different than Celsius. It's a whole other.
It makes more sense, like metric does.
But no, I've been looking it up for months, so I'm getting a little better at it. But the thing that I was looking at this, I go, gosh, there's times in the Central Valley where at night it doesn't cool off below that 25 C. So when do those flies go out? Or are they just groggy? Like they just get off of shift work and they're foraging tired and picking the wrong thing to eat, wrong tree again. Oh, what am I doing? I'm so tired. Like, do they take the naps or do they just suffer in the heat? I don't know what they do. That's somebody else should do that paper.
They die.
What if you leave the temperature on? But yeah, they're saying some parts of it could actually be a biological response, but they haven't found this circuit in humans yet. That's the next thing they want to start looking at is humans, and see if they can define this sort of circuitry somewhere within humans that induces that midday sleep on the hot days.
Well, you don't want to waste energy when it's really hot, and you don't want to lose water when it's really hot. And exerting yourself does both of those things. So if you can just shut it all down
and chill.
Yeah, that is extremely beneficial.
Absolutely beneficial to your physiology, your survival, resource use, all those things.
Yeah.
For sure. Hey, Blair.
Yes.
Did you want to talk about sneezes now? We've got hot weather survival and sneezes.
Why do we sneeze?
Because we have.
Oh yeah, I don't know. Because have you ever seen a pollen that does close up? They're all spiky and evil looking, and then you get in there and it touches your nose, and you're like, get out of here.
Get stuff out of our noses.
Yes,
exactly.
So itchiness or pollen might cause us to sneeze, but the reason us humans can sneeze is to expel stuff from our nose. It's to expel waste, essentially. Sponges sneeze also. some of the most rudimentary animals in the world, simple animals, simplistic body design, whatever you want to say. It all sounds derogatory, but they're basically just a living tube that lives in the ocean. And I love them, don't get me wrong. But it appears that they also sneeze to expel waste. Field experts have known about this kind of sneezing behavior for years, but they have not known the exact mechanism or the exact reason for this behavior. And so this recent paper that came out in Current Biology looks at it as a method to get rid of materials the sponges cannot use. Now, to be clear, sponge sneezes are not like our sneezes. They take about a half an hour to complete. They are a slow motion sneeze.
Because they don't breathe.
Right.
They don't have the big baffles to expel a large quantity, right?
So they're
All
kind
at
of
once.
just twitching to move, consolidate, and push away yuck. This is essentially what's going on. But both sponges and humans sneeze essentially to expel waste as a mechanism. We might be able to walk away from a place filled with dust or pollen. They cannot if the water around them gets too dirty. They need to find a way to clear out that water so they can continue to filter feed. And so organic matter exists in the water around a reef where these sponges live, but most of it isn't concentrated enough to kind of be in places where other animals can eat it. So it also turns out that by sponges doing this slow motion sneeze, they are concentrating and expelling this edible mucus in small, convenient packages, like little snacks for animals to eat.
Sneeze packets.
Yeah. And so they think that most, if not all, sponges sneeze based on this research. And there are potential future opportunities to understand the material cycling, exactly the mechanism, how it's moved, how they know when to do it, kind of like what the impetus for it is. And so it also actually looks in these videos like the mucus moves along predefined paths on the surface of the sponge before they accumulate. So they would love to go through, take more videos, try out different waste distributions in the water, check different species, do all this stuff to try to figure out what is happening with sponge sneezes. But for now, we know for sure they sneeze, and their sneezes provide delicious mucus to other fish on the reef for food. So they don't eat their own boogers, but fish do. So there you go. Sharing is caring.
Fada in the chat is asking you, are we sure this is a sneeze? Why isn't it defined as a fart? I guess it's because you can't smell the difference underwater. Maybe? I don't
know.
No.
And Gretchen Gamer asks, why don't we sneeze while sleeping? Oh, we do.
If you have,
we do all of the time.
Yes,
you just
usually
you don't notice.
So my understanding is that sponges have one opening. So they water comes in through the pores and out through the top. So this stuff is coming from the surface of the sponge. So I'm actually going to answer this question. If it were a fart, it would come from the top of the sponge where water comes out. Yes.
Right.
But because it's coming from where water comes in, it is more akin to a nose and therefore is a sneeze, let's say.
Because it's releasing mucus, and that's one of the big hallmarks is the fact that mucus is involved.
Yes, farts are not well known for mucus as part of the transaction. So this is definitely
you hope.
You hope. Not ideally.
Not ideally. Blair, thank you for this fantastic information.
So
now we can answer the question.
So next time you're washing your dishes with a sponge, just think about it. Think about it sneezing. Or other things we've talked about. You're welcome.
Or other things, yeah. Now I will be contemplating sponge sneezes and sponge farts for the rest of the evening. But we have more science. How about plant roots? Plants like to put down roots, but maybe they don't put them down exactly the way that we want them to, or the way that is the most ideal, or maybe learning about the genetics behind the way that plant circuits, plant genetic circuits, proteins that work together to define the paths that plant roots take. Maybe understanding those circuits can lead us to being able to implement genetic control over not just plants, but other complex biological systems. Yes. So a paper out in Science this week out of Stanford University and some other collaborators. Researchers have taken a look at plant genetic parts. These are plant synthetic biologists who are interested in spatial patterns of gene expression and have been able to engineer them in plants to actually modulate root morphology. They've changed the way that the roots look. So historically, yes, genetic control, we've been able to control all sorts of things, and we've learned about various circuits that work in a sense as logic gates. And specifically in this study, they focus on initially a species of plant that it's kind of like tobacco related plant, Nicotiana benthamiana, and they were able to develop and measure circuit performance using green fluorescent protein to be able to see where their circuits were ending up. And this circuit that they were focusing on, it's a logic operation known as a Nimply gate. And they were able to control this Nimply, spelled Nimply, this Nimply gate. They discovered that there are certain transcription factor proteins that are modular. So there are binding and regulatory parts that are physically and functionally separate. And what this led them to be able to do was determine a bunch of and/or Nimply gate functions in this tobacco relative. And then they took what they learned in the tobacco relative and they put it on a model species. You're talking about model species earlier. In the plant world, Arabidopsis thaliana is the big model species, and
tiny model species. Big model, tiny plant.
Yeah, but they were able to alter its root morphology. Initially, though, one of the interesting aspects of the study is that they discovered that it wasn't a direct correlation. And so half of the circuits that worked in the tobacco relative did not work in the Arabidopsis. And what that indicated to them is that there's a lot more complexity. And so Arabidopsis is a slightly more complex plant organism than the original that they were studying. And so this increased complexity changed the outcome of these logic operations. There were other things in the black box doing work. Eventually, they were able to determine what those factors were and get all of their logic gates working. But the whole idea is, with this synthetic gene circuits, is being able to put stuff in place to, hopefully, eventually, deal with needs for increased nutrition in plants, because maybe we'll be growing more plants on less ground. How are we going to deal with the nutritional needs of people? Maybe we need to get more plant matter out of plants for the populations that we have. How are we going to get these plants that we use to deal with climate change? Can we get plants to actually help us fight climate change? So on the plant side, synthetic biology and control over organisms could be really useful. But then the fact that they're doing this kind of stuff and learning about these logic gates in plants suggests that it could then be applied to other organisms with greater complexity. Mm-hmm. I'll just leave it to you to imagine where it could go. I don't know, Blair, maybe this is the kind of research that we're going to need to bring back things like the thylacine or the mammoth. Maybe these are the kinds of genetic controls that we're going to need for the rhinos, for the hybrid organisms that we're going to be potentially trying to save. Or, maybe this is the kind of stuff that's going to be necessary.
Wow. I love that idea. I'm sure everyone saw that somebody put out a press release this week that they're gonna bring the thylacine back.
Yeah.
Great. We'll see.
Well, great. Great.
No, I love this. This is really smart. Taking some of the limitations and just ditching them completely.
Yeah. I don't know. I think it's interesting also when geneticists and biologists start looking from a computational perspective at biological functions. So this combines with this. How does it work? We're going to have an IFTT if that, this, then that. That's what IFTTT. If this, then that kind of situation in biology, right? With genes.
Mm-hmm.
Yeah.
Man, this is like just the beginning. I feel like we're on the precipice of such a cool field of innovation with genetics that I cannot even imagine what we're going to see in the next 20 years. I'm so excited. We'll be here to talk about it.
Maybe we'll be talking about the return of the thylacine.
Oh man, that'd be so cool.
Dang, things are everywhere. How do we get rid of all these thylacine? What is it there? I don't even know what we're talking
Tasmanian
about.
tiger.
Oh, it's the Tasmanian tiger.
Yeah.
Okay.
Yeah,
There's that's the devil, the Tasmanian devil. Exhibit that's they're opening a Tasmanian devil exhibit at the Copenhagen Zoo.
Very nice. Yes, if they opened a Tasmanian tiger exhibit, that would be very impressive because they are completely extinct.
Completely extinct. Thank you. Thank you for that.
Except for the hopes and dreams of a few people with money and science.
So much money.
So much money. Let's talk about old things. Justin, you have some ancient genes from South America?
Oh, good question, Kiki. By looking at people today with the rare Q haplogroup, researchers have traced the roots of South American ancestry back over 19,000 years to South America.
Huh.
The study based on an archaeological study that compiled a data set of 1,661 radiocarbon dates. Like over 1,500 of them made on culture materials and from some 118 human teeth, bones, from 454 different archaeological sites in South America. What they found is that based on the diversity of Q haplotypes spread across South and Central America today, they must have evolved there over the past 19,300-ish years. So, and it could be much older than that, since we know there's a genetic bottleneck that occurs around 12,500 years ago, coinciding with the Younger Dryas extinction event, which places a barrier on which samples are even in existence today. So we're looking at post-bottleneck with the current modern human samples that we have with this Q. But because so when you have a haplogroup or one of these things, it's a big umbrella that broke off from a bigger umbrella at some point, and they have all these subcategories too. So from several different subcategories that broke off from this Q, they can backtrack how long it would have taken for them to evolve. And it also gives them, with the distribution between Argentina and Mexico, great confidence that all of this divergence going back 19,300-ish years would have taken place in South America, somewhere in that vicinity. So I did a little bit of further looking into it and found previous research that said the haplogroup Q originated in Asia around 20,000 years ago, only crossing the Bering Strait into the American continent sometime between 16,500 and 13,000 years ago. So now we know that is not correct. So again, the previous conclusion was based on assumptions about when the Americas were populated. And it almost seems like it just stated that and must not have had any evidence, must not have actually done the same sort of research or had this. Well, I don't know if those were based on backtracking or if that was from the Siberian sample, which was about 20,000 years ago. But anyways, it says, as we now have evidence of populations in the Americas much older than even the emergence of these genes in Asia, which is also not completely true, but the arrow seems to point in the other direction in terms of genetic flow. Not that anyone other than me, Justin Jackson, is saying this, but if they previously concluded gene flow based on data for time present in a region, and those data points flip in terms of which one's the old and which one's the newer one, perhaps also the conclusions they made based on that data should also flip.
Mm-hmm.
Perhaps. So there is, the overall estimation of the entire Q family is estimated currently to be about 30-something thousand years back. They, I guess, had the first evidence of it in Asia around 20,000 years ago. We now have pretty convincing evidence that it was in the Americas about 20,000 years ago. So where does it go back further than that? Well, in the Americas, we know people go back much further.
Well, we know now. We didn't always know.
For this gene, for this specific gene. But we also know now that we have the Americas were populated with evidence 30,000 years ago.
Yeah.
And so that's starting to be the time frame of the Q emergence. And interesting fact, the pre-Columbian haplogroup for native peoples in the Americas, 90% Q, or at least in South and Central America, with certainty. But there was that North American one, the 12,500-year-old child. That there was this big should they test it? Shouldn't they test it? And the Native American community allowed a Danish geneticist to go and do the testing. And I think they were Danish anyway. But they determined that kid was Q. And that's, I think it's somewhere far up in North America. So pretty much that's kind of starting to look like the arrow pointing the other way. It is extremely widespread, Q, though. It's a rare haplogroup everywhere else in the world. Larry David has the Q haplogroup, as do about 5% of Ashkenazi Jews. Sweden and Denmark have a similar 5%, making Scandinavia the highest Q haplogroup in Europe. And
then I have Q. I'm sure I probably have Q.
It's very rare. It's 5% of Scandinavia.
Oh, but it's in a lot. They've got lots of Scandinavia.
But that's the high. It's the highest. It's the highest of Q in Europe is in Scandinavia, right. And it's thought to have arrived there really early on, after Bronze Age times, 3,000-4,000 thousand years ago. It was also believed to have spread to Eastern Europe, the Middle East. Largely with the help of Mongolian hordes half a century ago. But it's really interesting in all these places it sort of pops up around the world. But in dominant pre-Columbian haplogroup of Native Americans, maybe north, but definitely south, it was approximately 90%. So a very, very strong signal of this haplogroup in the Americas and the Americas population is coinciding with how old they think that haplogroup is. So there's some figuring they need to do, but they probably won't come to. Yeah, go ahead.
Which if you think about the timing and all these things, this probably suggests a raft across the ocean from Asia to South America at some point.
No, with this, I mean, the thing that I'm
what is it supporting?
Right. The thing that I think it's starting to support is not the Bering. Well, it's probably likely to be the Bering crossing area. The Bering
Okay.
Strait, right?
Because of all
this
the
is an area-
northern.
well, but this is an area that horses went back and forth many times throughout the eons, right? It would at some points be an open valley you could walk across with grasslands, even going if you go back far enough. Earlier than that ice age, that that they thought that there were islands there, but still, there's times where that's a very traversable area. And the next place we see this cue and Native American ancestry down in even into Mongolia and the Altai is in Siberia down into Mongolia and Western China. So what it tells me, though, what the story seems to paint the picture of, is a much earlier group, and I would say probably over 50,000 years.
That's old.
Go coming to the Americas as people are populating Australia. Yeah, I would say maybe even older.
Yeah. We don't have that evidence, though, right? The physical evidence.
Yeah,
we have.
No,
we don't have physical.
This
potentially points that. Yeah.
But this is and then people coming back and populating Siberia after having populated the Americas. So that you go the other direction, that ocean you can't cross. So we got to turn around and go all the way back. Right, go all the way back over. And then that is reintroduced because there's a couple of bottlenecks in Asia where populations disappear, where there were cultures that we don't have explanations for because we have no human remains for, but we know we're not related to culturally to any of the populations that came in after. All of it starts to add up to paint these really strange pictures of a potential peopling of the Americas, much more ancient than we thought, and then a return to Asia to add that back to that gene pool, which is why the signal is strongest in the Americas and gets less and less strong as you get further away from it. It's why the timelines are older in the Americas than they are when you get further and further away from them. Like all of that does paint the picture that if it was flipped the other way, people would say, aha, here's proof and evidence of. But then you flip the data and people are like, well, we don't have any evidence that conclusively can tell us one way or another way.
We don't have it.
It seems like we're constantly faced with this conversation of like, but how much gene flow is happening? How much are humans moving? How much? And it's like, I hear conversations about this stuff and it sounds like when you talk about something like a tiger salamander that's a ring species where like you have very distinct genetic populations in very distinct areas and they really don't leave those areas. But really, it's sounding more and more like we were migratory birds. Like we were just all over the map, right?
So
like we were little sandpipers, just like
other places.
Nope, I'm going to cross a continent.
Yeah.
I'm going to cross an ocean. I'm going to see what's over there. Like I am not constrained.
But keep in mind, these are over thousands of years. So these populations themselves, even ones that are living, 100 miles from each other, may not meet. They can remain very isolated, forming specific languages, specific genotypes that don't mix for a very long time, which is partly how we are able to also tell that these disparate, isolated groups had to have gone back over a specific, 20,000 years to have been from a single source because of the lack of interaction for so long.
I don't know. I'm not convinced that they never that things were as walled off as you say, because also when you study animals, you always think like, okay, this is where this animal hangs out. And then you put telemetry on them and it turns out, no, you were wrong. They were all over the place. Like it's hard for me to believe that in the Americas, for example, that over the course of a few weeks, somebody wouldn't just go on an exploration and go hundreds of miles and run into other humans and then just be like, all right, I'm going to go back or whatever. But, it just, yes, I don't like the idea of these like very strict walls and divisions of.
No, nobody likes that. People. But Blair, go somewhere that's 100 miles from San Francisco without a car and don't buy anything.
Carry everything you need with you. You have to.
Tough.
You have
It's
to forage for your food. Yeah.
You
and-
do.
Sure,
And
I'm
the
not saying
weather's
it was
gonna be different.
easy, but I'm also
I know.
saying there was no TV, so
But if like Justin's saying, horses were going back and forth, other animals are traveling back and forth, the idea of nomadic human populations that followed animals as they were hunting them, as they were following. Oh, the rains are coming, the snows are coming. We're going to move from the mountains down into the lowlands, and we'll go from the lowlands and we'll go to this other area. Oh, look, somebody found this river. Let's go there and have fish. You could see how it could move over time, but yeah, the idea of a single person just like going on a walk. It happens, but it's not as successful.
But I'm sure somebody did it. Yeah, of course.
Sure. That's why we have the National Geographic Society right now. And
Greenland is that story. No, Greenland. The current native population of Greenland was from a Canadian native population. A Canadian or far north United States, whatever, somewhere in that area, who apparently committed a murder, and was like, totally convinced someone.
I'm gonna scoot.
Oh, I have a vision. There's this place way over here. We need to go there. I had a vision that says we're gonna go. So they all followed this guy, sledging, crossing water, makes it to Greenland. And the Inuits who are in Greenland are on the brink of starvation because they've been in isolation, lacking technology for a long time. They're like wearing muskox. And no, they're not wearing. They're wearing seal skins because they think muskox are dirty animals, so they don't hunt them or eat them because they've got some rumor that you don't want to be a muskox. So they're not wearing fur anyway. They came and basically saved the population, this murderer on the run, by just going on a long walk, hundreds of miles and crossing waters. It exactly happened. Yeah.
Time to make a move.
Good old Kana. Good old Kana.
Oh, spiritual leaders. Get away.
The eagle told me we need to leave town. Snap, everybody. He said, quick. That eagle said, we got to go now. Take what you got on you. Just fly like an eagle. Let's go. Come on. Keep your head down.
Fly like an eagle or like a styrofoam cup to the recycling plant. I'm going to switch topics.
Is styrofoam, has that been recyclable all this time and I've just been neglectful?
No, it's very expensive to recycle. It's not easy to recycle. I mean, really, for the large majority of what you're going to do with your styrofoam is it's going to end up in the landfill. But researchers at Virginia Tech University, also working with a colleague at Santa Clara University and Dongbei University of Finance and Economics, have published their paper in Proceedings of National Academy of Sciences, a new process that they say is relatively affordable and scalable to large situations. And the key here is it's not really recycling of polystyrene or what we officially know as styrofoam. Polystyrene, it's hard to kind of melt it and turn it into other polystyrene. It's like once it's in its shape, that's kind of where it's going to be and how it's going to be used. So turning it from one use of polystyrene into another use of polystyrene is expensive and difficult. However, these researchers have published a paper that in which they've taken chemical reactions where they have also involved ultraviolet light irradiation and been able to break polystyrene down to its aromatic compounds. And then by adding certain solvents, they could extract different materials. So they get diphenylmethane. They've also got raw products that can be raw gasoline, asphalt. They've also got 4-oxo-4-phenylbutyric acid, a whole bunch of other things that are useful in chemical operations that could then also be sold for a profit.
You say they did all of this in an RV on the outskirts of Albuquerque?
Yes, no, not that. And we're not talking about Breaking Bad. This is
That's,
this is Breaking Good. This is chemistry for good. They suggest in their paper that this process is scalable and that the estimated cost of building a plant capable of producing these products is a mere $1.28 million. Mere. They conclude that this strategy of upcycling is viable. It could get rid of plastic that piles up, the polystyrene, styrofoam stuff that we use for takeout containers and plates and stuff, and also give a financial incentive to take care of it because the products could be sold. Yeah. So fingers crossed that they, with this paper, are selling their idea of
I still want to see them add into their equation before this is a thing. The cost of my time of sorting another thing, and the energy that takes, and the fuel it takes to have a truck. That's because it's like you can fill up the truck with, several blocks full of aluminum cans. You get the cardboard truck, comes up several blocks. It goes through a whole neighborhood collecting the cardboard. And the styrofoam truck comes by and there's like one piece of styrofoam. It's got to go back to the factory.
So, think about like packaging in factories. There's mass styrofoam being produced for sure.
Yeah.
That could go to this besides your cup. However, what I was going to say was, I understand the effort here and I applaud the effort. However, why don't we just stop using styrofoam? There are places where it has been completely illegalized. Like in San Francisco, you can't use styrofoam anymore. In most of California, I think styrofoam is illegal. There's different packaging you can use. There's different takeout containers you can use. It's pretty much gone in my life, and I do not miss it. So I don't understand why we can't just cut it out, why we have to be doing this.
I agree with you for the personal products. I don't see why polystyrene is used in a lot of cases. We could just do away with it but there's a lot of it out there in the world.
Certainly there is.
Could be remediated. And
I would just say, let's not make any more.
I don't know. Why are they going to pack your $1,500 62-inch thin flat screen quantum 8K television?
That like papery, cardboardy forms. Or how about the styrofoam made out of mushrooms that are biodegradable? How about that?
That sounds like it. What Kiki?
I think you can take it and put it in the closet, and then you can grow the mushrooms in your closet. No,
Blair is pointing out is the first of the 3 R's, because you got
reduce.
Yeah, you got the reuse and recycle. That comes later. The first thing that just eliminates the problem altogether. Just reduce the amount that you're using. You don't even have to reuse or recycle anything.
Amen.
Yes.
This is This Week in Science. Thank you for joining us for another episode of Science News Discussion. We are so glad to be here with you once again. Thank you. If you are enjoying the show, please share it with a friend today so you can share that discussion with someone else. All right, some quick COVID news as we come back from that little break. What's up with COVID? Well, a new study has been published. It's a preprint, not actually published in a peer-reviewed journal yet, but researchers working together looking at 99 people with long COVID for the last couple of years. They've been following them for over a year and doing deep dives into their blood, their T cells, and their immune signals. So basically looking for signals within the blood to see what's different in the blood of people with long COVID. Most of these people were struggling with intense fatigue, brain fog, and other symptoms. They seem to have a common factor, which is low levels of cortisol. which is a hormone that helps in controlling inflammation. They had glucose differences, and this is also involved in their sleep cycles as well. And features of their T cells suggested that the T cells were struggling, that they had been working very hard, and they were still working very hard, but they were tired. So there are particular signals that T cells give off that they are working, that they are overworked. And this study falls right in line with another paper that was peer-reviewed back in January that did document also low cortisol in people with these long respiratory symptoms. The big difference being number of subjects in the two studies, and the initial study only followed long COVID patients who were about 3 months after infection, where these were looking at people who had been recovered for many, many, many more months. They actually found it challenging, the researchers say, to find people who were fully recovered from COVID. Many post-COVID volunteers said they were healthy, but then when it came down to it, they were exhausted during their gym workouts. So the numbers of people they actually ended up recruiting were less than they were hoping to get. Anyway, long COVID probably has to do with chronic inflammation, probably has to do with these exhausted T cells, the virus kind of sticking around, similar to stuff like Epstein-Barr and chronic fatigue syndrome, and herpes virus, where they can sit and be dormant inside cells for extended periods and then kind of resurge. And so SARS-CoV-2 may also be something that lingers in some patients as well. The highlight of this, good news though, is that it could be possible to use this information to start targeting therapies that address the decrease in cortisol, that address the depleted T cells, and start treating it kind of as an autoimmune disease to calm the immune system down. So potential benefits.
This is like the withstanding mystery about COVID, right? Is the long COVID situation, and it just does not want to go away in some people, not everyone.
Yeah, small 20 to 30% of patients.
That sounds like a big percentage to me.
I'm
wondering how.
Yeah.
Well, I'm wondering how many other ailments are long haul ailments, and we
that's
don't
interesting.
know. Because this is one of the few times in all of our human history where we all got the same thing at once. Well, I didn't get it.
And we started having the science, the technology to be able to track stuff.
And the technology to track it. But it was sort of like the anecdotal women were talking about, after the vaccine, like their menstruation cycles were a little bit different than they were used to. And then they did a study and they found out, hey, that's not just an anecdote. That's not just a one or two off. That's a thing. That's a trend that we connect with the vaccine.
And that's the same thing that was long COVID as well, because people started using social media to connect with each other, saying, hey, my symptoms aren't going away and my doctor isn't believing me, and and they say it can't still be COVID. And so there was a real surge of people finding community online as well, and that enabling a groundswell of enough people, enough voices to actually make researchers really take note.
Yeah, and I think to Justin's point about how 20% actually feels like a lot, I think that's an important piece of the conversation that again and again is not being talked about when we talk about, like, oh, well, you're vaccinated, you'll probably get COVID now, but you'll just get it for a little bit and then you'll be fine. You'll just miss some work. It's not a big deal. Like, open everything up. This is the piece that I feel like keeps getting ignored, is if you have a 1 in 5 chance when you get infected of having this for months, that is a different risk than just being sick for 5 days.
Yep. But if we don't talk about it, nobody thinks about it. It's okay. Yeah, and and these numbers may also be I was going to say these numbers may also be there is conversation about these numbers because different numbers have showed up. It's been as low as 8%, 5 to 8%, as high as 20 to 30%, depending on the study and depending on the sample of individuals that have been looked at. So
depending on whether or not their doctors believed them.
I mean, that's part of it, the data collection, right?
It's just funny that people think that they can win the lottery, which is a 1 in several million, but they don't think that they're going to get a disease or the effects of a disease that, even being optimistic, are around 1 in 10.
Yep.
Oh.
Well, speaking of data collection and communication and who we trust around things like COVID, the news this week is that Rochelle Walensky, the director of the CDC, has ordered a lot of reorganization.
Shaking things up.
Shaking it up at the CDC, restructuring things, really taking to heart the fact that the CDC failed when it came to giving guidance to the United States population, that COVID itself was, and the guidance they gave was confusing and overwhelming. She has brought in people from outside the agency and also is doing agency conversations, talking to people working in the agency. They're basically doing a big review to see how the systems in the CDC work, what's working, what's not working. They're going to be restructuring where scientific agencies report. The scientific divisions in the agency are now going to report directly to Walensky's office. There's going to be a new executive council reporting to her as well, setting the priorities for the agency and directing spending. And then as well, when it comes to spending, there has been an issue for a long time regarding something like a pandemic for a one-time big push that involves the entire United States and coordination across many states. The funding the CDC has really isn't there, and they have to go to Congress for special funding to be able to fund the work that they do. And previous CDC directors have actually said that, they didn't have enough money to send people on flights to go to places where outbreaks were happening, to be on the ground. So there are issues related to where the money is, how much money they have, how that money is being spent. Additionally, the CDC has to come back from a lot of the changes that occurred during the Trump administration. One of those being a complete shift.
Response team.
The pandemic response team.
Right.
Yeah. And additionally, during, it was in June of 2020, it was announced that there was a company that the Trump administration directed hospitals to stop reporting all their data to the CDC directly and give the data to a Pittsburgh-based software company called Teletracking Technologies. The company had really never worked on anything this big before. A lot of people were like, "What are you doing?" But it came out that this company was very closely related to big funders, owned by funders of the Trump administration.
You don't say.
Yeah. And so there was a lot of trouble getting the data from the hospitals, then to the teletracker, then to the CDC. The data was getting sifted and edited, and there were pieces in the puzzle that did not quite fit anymore. And so, the CDC couldn't give good guidance because they weren't necessarily getting the data that they needed. So as of December this year, that contract is ending and all data reporting is going to go back to the CDC. And so they're going to be really looking at how the data is being put. Yes.
Grift, the gift that keeps on giving. Well,
the grifting.
Yeah, and the other thing too, in all of this shake-up and doing things better too, like, there's a point when it's just not your fault. When you are telling people point blank, we need to take XYZ actions, and a big part of our population says no. You've done what you can. If the lemurs are still going off the cliff,
Lemmings. You're thinking of
well,
lemmings.
lemurs. I'm sorry, the lemurs. Lemurs,
And the lemurs too.
You would
Many of
think would
them.
know better. You would think they would know better, but they get caught up in all the excitement of the lemur. Oh gosh.
The lemmings.
What's the thing? Lemming.
They don't even live in the same place.
When you're throwing
No.
lemons off a cliff.
Yeah. Thank you.
Llamas.
Thank you. This is how Blair got the job. She could real time correct me. But they were giving
Yeah.
at some point got to good advice that needed to be followed. Previous to that, I think we would agree. We were seeing scientific publications we were talking about on the show that made it sound scarier than the CDC thought it seemed to stance was anyway, or even the World Health Organization's stance at that time was. But we were seeing studies coming out saying, "Oh nope, this aerolizes. It stays in the air. This is respiratory. It's not about touch." And it took a long time for them to adopt the findings that science was making. No, I noticed that in real time as we were doing the show, but there was the point when they're up to speed and they're giving the good advice, and that's the majority of the takeoff of the virus spread is when people were getting the right advice. There wasn't a messaging problem from the CDC. There was a dissemination problem through media. That's a whole other issue that's out of their control. So
Agreed. A dissemination problem. And then also, communications really comes down to the understanding when you have a message at hand and how people are taking that. And so there were big gaps in making sure that real understanding was happening as opposed to just, this is our message. Yeah.
And most importantly,
I was just going to say, props to Rochelle Walensky. I mean, she is accepting they did a really bad job. They've dropped the ball on monkeypox as well. We are not in a good position, and she's taken that and she's saying this needs to change. It's not reflecting well on anyone. Who knows if trust can be redeemed, but they're going to try.
Yeah, and we've bought like 60,000 doses of monkeypox vaccine that are still sitting in Denmark because nobody's come to pick them up.
What?
Good.
Yeah.
I'm sure they'll be fine.
We paid for it. Apparently they ship fine. They don't need to be cold temperature stored, I don't believe. But yeah, they're sitting here in Denmark still. The company's making them, just filling up a warehouse. America's paying for it, and still haven't come to pick it up. But I did want to get to one important thing before we go because I just heard this.
United States federal government, you have a shipment to pick up. Is anyone going to come by here? I mean, I know it's like a weekend, but do you want to come pick up these monkeypox doses?
It is confusing to pick up a package in Denmark because it could be it could be anywhere. You could go to a 7-Eleven and pick up your packages. Like they ship it to all these weird mom and pop liquor stores. No, it's like really weird. Or you have a code on your phone and go to a drop box and a grocery, like in a parking lot down a dark alley. Like it makes no sense. Sometimes you have to go across town, pick up packages at different places. It's ridiculous. But I did want to point out, because this is very important to finish this. According to something I just heard from Beach Price in our chat room, lemmings don't go off cliffs either. We all stand corrected.
So much. I just want all the things that start with L.
Yes, JG, when life gives you lemurs, make lemonade.
When life gives you lemurs, trade them in for a llama. This is TWIS. We hope you are enjoying our show so far this evening. Thank you so much for joining us. If you are looking at this show or listening to this show and thinking to yourself, I wish I could help this show out. How can I help this show out? Well, first, make sure you click a little heart, a like, give us a little like somewhere in there so that other people know that you like it. Subscribe and tell your friends. But the big thing is that you can go to TWIS.org and click on the Patreon link. Patreon is our listener support community, and anyone who chooses to support us at $10 or more per month will be thanked by name at the end of the show. I'd love to read your name. So head over to twist.org, click on that Patreon link to help support TWIS now. We can't do this without you. Thank you so much for your support. All right, let's come back right now to the part of the show that's going to be full of Finch brains. Meep, meep tonight. It's Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Well, at Kiki's behest, I suppose I should start with some Finch brains.
Always.
This is a study from Okinawa Institute of Science and Technology. Had a lot of studies from them lately. They are on fire with their animal science. They wanted to look at how juvenile zebra finches learn songs directly from another finch. For the purpose of this, what we call a tutor. This is usually their father, but it can be other older birds. And that social interaction keeps them motivated and on task to learn songs. This study wanted to know how those things are related, whether they can learn songs from a speaker, if they can learn songs from a recording of a tutor, or if it has to be a live present tutor, and what is going on in the brain if it's dependent upon a live teacher. So you guessed it. If they are only hearing from the speaker, it doesn't really help. They do a pretty poor job learning. It has to involve a live tutor in some way. So in the study where they wanted to look at kind of first of all what it required for finches to learn quickly, over a few days they recorded brain activity and under the following situations. The first was with a juvenile who had never heard a tutor song before, listened to it through a speaker. So that's like the least connected. Then we have a juvenile directly interacting with the tutor as he sang. And then finally, they went kind of in the middle. They listened to the tutor's song through the speaker, a tutor they had already met. So they found that generally speaking, they needed some sort of social context. They needed to have a tutor involved in some way to be able to learn the songs. It seems like they did best with the live tutor, but they also did very well if they had already met the live tutor and they were listening to that individual through a speaker. If they were listening just to an unknown bird through a speaker, they didn't do a very good job learning. So they wanted to know what was going on in the brain. And this is where I'm going to do my very best and Kiki can chime in and help. So this all started with about 20 years ago. Scientists studied human babies and found that they needed a personal guide to recognize pieces of language. And so their idea was that there was something going on in the brain that cued them in to pay attention. It's all a question of attention. Store this data. Pay attention. Don't look elsewhere. Listen to your tutor. So they focused on a brain area called the locus coeruleus, LC. Did I say it wrong? Say it. Okay. All right. Which is known to be involved in attention and arousal. And so neurons from this brain area, they helped them to kind of order their auditory functions. And then they also wanted to look at the caudomedial nidopallium, NCM. And so they, from previous work, have decided that this is where juveniles' memories of these tutor songs are formed and stored. So they wanted to look at these two areas. Their hypothesis was that these two circuits working together might be important but they hadn't looked at juveniles in particular and didn't know how having a live tutor there related to that. So both brain regions responded more strongly when the tutor sang, and they heard the tutor singing through the speaker. That also lit up these areas of the brain. They responded well. The LC neurons were active constantly while the tutor sang. So they were responding to the vocal communication itself more than specific notes. It wasn't like peaking at certain moments in the song. And if the researchers inactivated the neurons from the LC to the NCM, so this circuit between these two areas, when the tutor sang with social interaction, they were unable to accurately copy their song. So they think it's this area of the brain for sure that is kind of turning on and lighting up and kind of logging the information. So the LC, they think, is conveying a secondary type of information. They're not just saying, hey, listen to this music. They think that the LC is focusing attention.
Mm-hmm.
So the NCM neurons are actually cueing into a bunch of inputs from different regions in the brain. And the LC, in connection with that other area of the brain, is actually what's telling them, hey, this is important. Learn your songs.
Right.
Does that all make sense to you, Kiki?
Yeah, it makes a lot of sense. Yeah.
So it's,
Yeah.
you know,
The bird brain, we've known about this circuit for song learning and song production for a very long time. And it's been this kind of analog to human vocal learning for, like you gave the history, but understanding exactly how that tutoring aspect works and why a baby bird doesn't necessarily learn from just anybody. How does it learn from its parent tutor as opposed to the male that is, the closest other male in the colony? These birds are very social in nature, so a specificity would be very important. And so having that kind of attentional focus on the most important caregiver
Mm-hmm.
or
Mm-hmm.
picking, a particular individual to be the tutor would make a big difference.
Yeah, and so you're actually cueing into exactly what the next step of this research probably is going to be, which is the cues that juveniles are using to capture social context and identify the importance of this one individual. It's exactly what you're talking about. What cues are telling their brain, these are the songs you got to pay attention to, listen to them, copy them, store this away for later. This is so important to the development of these little finchies and when they're forming their memories of songs. So they want to continue to investigate how social learning circuits function, which is what are the cues of this individual? Like I said, is it like a chemical? Is it other information that is lighting up these particular neurons to gauge the importance of which zebra finch is important and what they're doing is important? And so they also want to see this. I didn't even think about this, but they also want to look forward into the future to see what cues the juveniles give the tutors to encourage the tutor to teach them. Because
that's a good one.
Yeah, it also appears to be more than, it's not passive. It's like they're saying, like, repeat after me, and they're singing, and so there appears to be
no toodle doo
exactly
toodle doo closer.
Read to me, sing to me, right? So how are they telling their tutor, like, I'm ready to learn, sing for me so I can learn? So there's a lot of dynamics here we're not totally understanding, but I think the thing that I really liked about this is that there's something going on in their brain specifically telling them, hey, listen up, this individual is important and this information is important.
Yeah. One interesting thing about the whole song system there as a sideline is that these birds are kind of born with a little bit of an idea of what a song should sound like, and then they listen to their tutor, and then they do a little compare and contrast, and they have this like plastic period after they leave their tutor behind to figure it all out before they head out on their own to become
Get some
an adult.
individuality, perhaps. You know?
Yeah, and they have little slight variations of their own, but it's the tutor helps mold and shape the song, which is very stereotyped within a bird like the zebrafinch.
I love it. I think
So cool.
in that tiny little bird brain, there's just so much going on.
So much, yeah. And then you go from there, and it's like, okay, in the bird brain, you've got this attention. Oh, pay attention. This is the thing that you have to listen to compare against to make sure that you're getting the right song. But then in other birds that are social in a way where their vocal system is more plastic, do they have that same kind of attentional aspect? Like, so that's that's a question. If the song isn't so stereotyped, if they learn multiple songs, if there are, like in parrot species, the brain is a different beast in parrot species, but
totally
they have a very different social structure as well, with something like you might compare to a chimp social structure with multiple adults that kind of teach you with multiple tutors. So very interesting questions that can be delved into from there.
Yeah, and then you have bird species that have really basic, boring songs. Like, how many times do you have to hear a peacock before you know how to be a peacock, right? Yeah, really. Anyway, moving from tiny bird brains, let's talk about tiny spider pedipalps, huh?
Okay.
Yeah.
Let's do that.
So this is a study from the National University of Singapore, and they wanted to look at a specific kind of theory about how males can combat female cannibalization. So if you
oh, do tell.
Yes.
How does one avoid being devoured by the female of this species?
Running away. I mean,
yes.
maybe other than.
First instinct.
So there's evolutionary arms races that happen between species, like predator and prey. But there are also evolutionary arms races that happen between male and female when there is an intense size difference, and or the female often tries to eat the male during or immediately after copulation.
Oh gosh.
So there is a evolutionary advantage to a male who can either escape being eaten, because then maybe he can live longer and father more offspring, or if he can just find a way to father more offspring despite being eaten. Similarly, if the female gets a benefit from cannibalizing that male, like, for example, nutrients, sweet nutrients, that allows her to grow more eggs and have better quality offspring, then there is an evolutionary advantage for her, right? So there is this push-pull that's happening. So there are different theories that kind of feed into this, and how specifically the males can kind of counteract the negatives of being cannibalized in a mating situation.
Stop tasting good.
I mean, they can't help it. So there's a theory of male mating syndrome, which posits that male spiders are under sexual conflict pressure in sexually cannibalistic situations. Yes, sounds correct. As they may only have a single chance to mate. So how can they optimize that single opportunity to mate? And in this study, they explored whether male spiders use additional cannibalism countering strategies by focusing on two mating tactics. One, the better charged palp hypothesis, which predicts that male spiders will selectively make use of one of their sexual organs, yes, they have two, known as pedipalps, that contain more sperm. So that if this is going to be their only opportunity to impregnate a female, pick the one with more sperm. So it's the better charged palp hypothesis. The other hypothesis they wanted to test was the fast sperm transfer hypothesis, which is exactly what it sounds like: accelerated insemination so that they can get more sperm out of their body before they're eaten.
Okay, okay.
Yes.
So, yeah. Question about the charged pedipalp
Yes.
hypothesis. If they're choosing the one that has more sperm in it already, that means that they are increasing the competition that their sperm are under when it comes to inseminating any spider eggs in the future.
Yes, although she has a lot. So I think the expectation is that more sperm, more fertilized eggs.
It's all good.
Okay. We have a picture up on the screen being shared there.
Yeah.
Is this the same species,
Yes.
male and female of a spider?
Yes.
Okay.
Yes.
Well,
The male is very tiny.
So tiny.
the male looks like
Food.
looks like a little cranberry, and
Yeah.
the spider there looks like
Looks
a
like a
giant
Yeah.
death spider, almost like a wasp.
Yeah,
with
so this
acceleration
is
on
an
that
orb
one.
weaver. Yeah.
So
Yeah.
with that size differential and looking like a snack, I mean, good luck.
Yes. So it's interesting you bring up the size differential. They performed comparative tests on 5 species of orb weaver spiders from Singapore. They had varying levels of female sexual cannibalism and sexual size dimorphism. They wanted to look at both. The proclivity to eat the male, and also just if they're just so much bigger. So they found essentially both of their hypotheses were true. Male spiders choose one of a paired sexual organ with more sperm for the first copulation with a cannibalistic female, and male transfers more sperm if a female is cannibalistic or when the female is of a much larger physical size. So they really saw that both of these are true. So this provides credibility for the male mating syndrome. Why do we care? This opens new research questions on the ability of male spiders to differentiate sperm quantities between his palps. How does he know which one? It's such a teeny tiny volume difference. It's not like, oh, one's heavier. I'm dragging on one side of my body. No, it's very, very small, the difference. So how do they know? So this is really the question now, is if they are really making that decision, they have to have some sort of signal, chemical, hormonal, I don't know what. Is it always the same one in a particular species? We don't know. So that's a new interesting avenue of research to look at. And then they also want to look at the mechanism by which they can just somehow transfer sperm faster. What is happening in their body that allows that to happen? So there's lots of future research that can come with this. But they also think that it's worth looking at just other types of spiders. They were only looking at orb weaver spiders, and there are lots of cannibalistic spider species. So
probably lots of different tactics.
Great. So are these two tactics happening in other spider species, or are there additional tactics for combating female cannibalism? Because this has been going on for millions of years. And so chances are lots of things have popped up in the evolutionary record. And it would be interesting to see what has been so far successful in being able to propagate as much DNA as possible before being munched up as a post-copulatory snack.
And if I've heard you right, they give more of a donation, I'll say, the more cannibalistic the female appears to be. So I'm not kink shaming any spider, but it seems like then a self-fulfilling destiny, if you will, that
of
they
the
will
cannibalism and the delivery.
If that's what the female spider wants, then he's encouraging her bad behavior.
So that's the question. Does she want more volume of sperm? Because She might not if she has multiple copulations with multiple individuals throughout a mating season, let's say, right? That it might not matter to her. She might get all of her eggs fertilized no matter what. Doesn't really matter.
Okay.
Mhm.
So that's
It's
really good
definitely,
question.
though, the survival, the males that survive, the tactics that they use are potentially the tactics that go on to survive. And so larger deliveries
Yes.
and whatever tactics they happen to be, looking for the bigger palp or not.
Yes.
And so, but then also,
Interesting.
isn't it like all of these must be naive little male spiders?
Some naive,
Like
some not.
the females be like, oh yeah, I've eaten several males this season, and the little guy spiders, oh, I've never even seen a lady before. Wow, isn't she pretty? Oh yeah, let's talk a while, shall we? Here, try this.
They're
What's
just
this?
under
It's wine.
hormonal control.
There's not
Don't even
alcohol
worry
in this,
about
is there?
it.
Oh
There's-
no, sweetie, you'll be fine.
You'll be fine. Some males do survive, though, and that
Yes.
is the interesting tactic as well. That's
I
why
will-
this is interesting.
They just
I
The
will just
males
throw
survive
out
do
once.
there-
survive.
They survive once, okay?
Or more.
You think they're surviving a second time? Oh no, I wouldn't bet on it. They just
This
got
reminds
lucky.
me of one of
Where the
my
phrase
very
comes from.
first one of my very first stories I brought to the Animal Corner over a decade ago now. Was about this, but in black widow spiders, and how males will avoid cannibalism by tearing off their own pedipalp and leaving it inside of the female. And we had a whole conversation about whether that would be beneficial to him or not, because if he can't copulate anymore, then there's no benefit to him surviving evolutionarily, right? So, so there's a whole theory that it's because it provides a plug so that other males cannot then copulate with the female.
Yes.
But,
and it also, but that
And
also,
then that would be the sperm competition.
May
aspect.
underscore that spiders do have a much deeper inner life than we imagine in those extra days on the planet.
Sure. Yeah. Eating some more mosquitoes, doing good work. Anyway, spiders, their pedipalps, and sexual cannibalism. Fascinating. It will not be the last you hear from me about this, and I cannot wait for more research. And that is the Animal Corner tonight.
Thank you so much, and we are looking forward to many more spider mating stories, cannibalistic spider mating stories to come in the Animal Corner. Justin, what kind of stories do you have for us right now?
Oh my goodness, I don't even know. Okay. So there's like this weird thing. There's this weird thing that it's actually a big percentage of the population. It's something like 20% of people have this strange response to the sound of somebody else eating or smacking of lips, that sort of thing. Researchers have identified parts of the brain involved in here as misophonia, a condition associated with an extreme aversion to certain sounds. The results from Ohio State University scientists suggest that one popular explanation of what causes misophonia. We're just intentionally turning off 20% of the audience. Intentionally. Listen, you don't know what you're doing. Individuals with misophonia feel anger, disgust,
Oh, I'm sorry, everyone.
I'm so sorry.
and a desire to flee when they
Oh no,
hear
we're done.
these
I
sounds.
promise. I won't do it anymore. I'm sorry.
Chewing, eating, lip smacking, similar mouth noises are most often associated with the condition. Previous studies suggest that misophonia is caused by supersensitive connections between the brain's auditory cortex and the oral facial motor cortex areas, those related to the face and mouth. This new study, for the first time, examines what happens in the brain when people tapped their fingers repeatedly, which is another sound that can trigger some people with misophonia. I'm picturing the clicking also of a pen, like some people I know.
That kind of sound could wear on you.
Yeah.
For sure.
But they found that the patterns of the brain connectivity when finger tapping regions were different in people with misophonia compared to patterns of connectivity with chewing regions. So what they're basically saying is that, well, they basically found in doing these fMRI studies is that it's not the motor neurons or motor cortex neurons, whatever, of the oral facial area itself that are recording this as well. So study involved, it's a small sample size, 19 adults who had fMRI scans in their brains while they performed various tasks, all completed 3 questionnaires that measured their level of misophonia. Based on these results, levels participants ranged from none to mild. They had tasks where they vocalized syllables, and it showed which regions of the brain were activated by speech production, which overlaps considerably with the oral facial movement, and thus is connected to sounds like chewing. Participants also tapped their fingers on their legs repeatedly in a separate part of the experiment to make another movement connected with misophonia. In addition, participants were scanned with MRI when they were doing nothing. Results showed when they were at rest, participants who scored higher for misophonia did show stronger connections between auditory cortex and motor control area, just as the previous study had shown. But when participants were actually using their mouths to produce sounds, a different region of the brain was active, and this region showed no stronger connections in those in high misophonia when compared to those with low. So what previous researchers identified as the oral facial region, they're involved in mouth and face movements, may not actually be the oral facial region at all. There was no connection to the auditory cortex at all. The important connection was the insula.
The insula.
Is that a part of the brain, Kiki?
It is. It is.
Oh.
The insular cortex, the insula. Yes.
Yeah, so it says this provides additional evidence that misophonia isn't about chewing and other mouth noises at all. It's just that those noises are triggering a part of the brain that people find really annoying. We have actual evidence in the brain of people that dislike sounds that aren't from
the
just
part of the
mouth and
brain.
face.
It's just triggering the annoyance part of the brain. It's just the wires go straight there. Doorbell, doorbell, doorbell.
But I guess this, what was it called? The insular cortex?
Yeah, the insula.
Is linked to strong emotions, including disgust.
Yes.
Is thought to be sourced there. So that explains a little bit, at least, of why some people are really annoyed by this. Hey, did you know that sugar isn't bad for you? But it can kill you.
What?
Yes. Well, that's because, like anything else in life, moderation is the difference between something that is harmless at one quantity and could be dangerous at another. High consumption of sugar. Oh, yeah, you can die from over drinking water. Absolutely. Yeah. It's also called drowning. Yeah. Also a way to do that. High consumption of sugar has been linked to an increased risk to a host of health issues. Food and beverage companies want you to feel like their products are safe and label products with reassuring claims about sugar content, like without added sugar or less sweet. Consumers rightly interpret such claims to mean the product is healthier than it actually is. There's another type of labeling, one unlikely ever to be seen in America, because for some reason, labeling foods is so wrought with lobbyists in America. But the type of labeling is called Nutri-Score, which is showing up on European products to inform consumers about the overall nutritional quality of food products. Previous research shows that the Nutri-Score can indeed guide consumers to make healthier food purchases. Kristen Jürgenbeck and colleagues at the University of Göttingen, Germany, published a study in the open access journal PLOS ONE examining how the Nutri-Score fared against typical, sort of call it vague or implied claims in advertising for sugary products, or about the sugar content, I guess. Advertising sugar claims. They conducted an online survey of 1,103 German participants. The survey asked participants for their perceptions after being presented with images of the packaging of 3 hypothetical products: instant cappuccino, chocolate muesli, and an oat drink. Oat drinks are a thing.
Yes, they are.
Yeah, is it just oat milk?
Yep.
Yeah.
No, it's not oat milk. It's not oat. It's oat. It's an oat drink.
Yeah, you can't call it milk. I know. What is
Oat
it?
here's
juice. Oat water.
Oat water. Yeah.
I'll read you one of the oat drink ads. When you don't have time to sit for an oatmeal breakfast, try oat drink. Like a warm bowl of oatmeal you can drink on the go. Just the thing for busy moms, dads, and children too. Oat drink. Made from real oats, now with less sugar added. That sounds healthy, doesn't it?
Sure.
Survey results suggest that when a Nutri-Score was not present, claims about reduced sugar were successful in misleading participants into believing that hypothetical products were either healthier than they actually were or better for you. I guess that's the same thing. When Nutri-Score was listed, when that was added to the same labeling, it reduced the misconceptions about the less nutritional food. On the basis of these findings, researchers called for restricted use of sugar content claims and similar labels, as well as the mandatory use of Nutri-Score by companies that do make sugar claims. They also call for future research to evaluate the effect of Nutri-Score for additional food categories and in the context of other advertising claims that could mislead consumers about food healthiness. Authors add, nutrition or taste claims about sugar on the front of packages can improve the health perception of foods with poor health images.
Yeah, it's like when red vines or some other red licoricey type, candies say now with 0 fat. This is a non-fat food.
Yes.
It's like, it is, but the nutritional content of this food is all sugar. So
all that sugar is going to turn into fat.
Right. And so if there were a Nutri-Score involved, that would affect the way that people actually take that non-fat food and take that into consideration. Yeah. We need Nutri-Scores here. Why don't we have Nutri-Scores? Why is it so hard to read a nutritional label? I don't understand.
It's on purpose. It's on purpose. It's on purpose.
It's because if you score food, if you score food, then you'd be giving a fair or you'd giving an unfair advantage to certain foods, and you'd.
No, that's
unfair
not what
advantage to healthy foods.
That's what they're concerned. The thing is, sugar is addictive and it triggers a reward system. So by having a lot of sugar in your product, whatever it is, people will buy it more because they got rewarded by the sugar drug system in your brain. And so they're going to and if you eat out where there are no labels, if you go out to some restaurant, there is so much salt and sugar in that food, unlike any meal you would prepare at home. Because they want you to enjoy your meal, so they just feed you full of sugar for that reward system to trigger.
I think it depends on what you're ordering at a restaurant, Justin. That's a strange one. The processed foods are the things that are chock-a-block with salt and sugar and other.
I bet you. You take your healthy night at a restaurant, ordering a pasta dish, say, or whatever the heck it is. Guarantee you're gonna have way more salt and sugar in that thing than if you made it at home. I don't know how to prove it though, and that's how they get away with it.
Way more. Moving on the heels of Justin's story here about the reward system, sugar, and nutritional information. And his story previous to that discussing the influence of the insula in misophonia. There's a comment in the chat room here that JG says there are a bunch of cool studies on how the insula plays a role in consciousness, that the anterior insula is called the gateway to consciousness. Well, the question still is, what is consciousness and how do we study it? We know we are conscious, because, we have that saying, I think, therefore I am. And the fact that I can say that statement and understand it and know that I understand that is something I'm saying and that I understand it, and I can have an internal understanding of my experience and my own inner world. I know I'm conscious, but maybe Blair and Justin are robots. Maybe Blair and Justin are artificial intelligences. How do I know that they are conscious? Because they tell me they're conscious, right? Are you conscious currently?
Maybe.
You better believe it.
Maybe.
Or maybe I'm a robot with a recall on the run.
On the run. Well, some researchers this week have published their work in Frontiers in Psychology, which they hope brings together the worlds of physics, biology, and philosophy in the study of consciousness. And they have, in their paper, suggested that we need to address consciousness in the study of it as a relativistic phenomenon. So, in the same way that that we've talked about Alice and Bob in many situations, Alice is standing on a platform, Bob is on a train. The train rides away. From Bob's perspective, he's sitting stationary on a seat, and Alice is moving away. But from Alice's perspective, she's standing stationary and Bob is moving away from her. So we approach a situation in this in physics with relativity. You have these two systems that are acting, that have actors acting relative to each other. So can we take that math? Can we take that understanding to apply it to the idea of consciousness, where we can only measure it through a related phenomenon of neural signals? I can measure Blair and Justin's brain signals potentially, and I can ask them questions about their consciousness, and I can read all those wiggly signals on a EEG printout or on a computer screen. But how do I know they are really conscious? This is just, it's a step removed. It's a representation of their consciousness. And so the only way that we can start to address consciousness and the real problem of consciousness and finding it within the brain is to look at it as a physics problem of relativity. You are relatively conscious compared to me.
I will.
So how do we
Justin is.
measure animal consciousness then?
Right. So the hope is that is the great question, because if we can start looking at this from this frame, we can potentially start seeing the most basic levels of consciousness and start defining what neural signals really do connect to consciousness, and then potentially start moving to different species and being able to, find signals that are very similar in other species as well.
I'm going to break the fourth wall here just for a moment, and I will admit candidly to you, you know who you are, because you're the one listening.
Yes.
You are the only conscious being on this planet. The rest of us are figments of your imagination.
Or are we? Right?
Oh yeah, of course we're not. No, we're all real people out here in the
fine.
world. Yeah, sure. Yeah, we're not. Oh boy, yeah, no. We're all other real people. It's not just all in your head.
According to their abstract, they say phenomenal consciousness, like a phenomenon, is neither private nor delusional, just relativistic. We're not figments. We're not delusions. We're just relative to your observation. Yes. And it all has to do with, the third observer, an outside observer situation.
As soon as they start doing that, then they're like, oh yeah, that's because in quantum, that's this. Stop it. Stop trying to connect consciousness to quantum things that you're misunderstanding in the first place. Everybody who even understands what's taking place in the quantum third party observer act will tell you that it's not anything that any of you think it is. It's just you. We're all figments of your imagination. None of this is real.
It's really, I mean, in a sense, it is true because our brain constructs everything in our living experience for us.
Mm-hmm.
So we are all a construct of our observers. Anyone observing you are a construct to them. Everything is a construct to you. To my brain,
a bunch of ticking clocks.
Yes.
Yeah,
real?
there's no guarantee that the light waves that hit your eyes are interpreted by your brain the same way they're interpreted by my brain, or the same smells are interpreted the same way, or any of our experiences are interpreted the same.
Right. And so that's one of the big issues with how do you find the neural trace of consciousness in a brain? Because every brain, even though there are big bits of it that are very similar, they're like a fingerprint, right?
Mm-hmm.
The whorls and everything. My neurons are not going to be specifically connected in my brain the exact same way that your neurons are connected in your brain. I've had different experiences in my life than you have. What my experience of happiness is not necessarily your experience of happiness. And so if I say, I know I'm conscious because I'm happy, and both of you say, I know I'm conscious because I'm happy, and then we go and we look at our brains and we go, what is happy? What is the neural signal of happy? You're going to find different neural signals. There will be commonalities, but they're going to be different. So you're going to lose resolution in the search for the conscious trace within happiness, right?
Yeah,
and also,
maybe we're better off not knowing.
As we've discussed too, if if we swapped brains, however that's possible,
Right.
or had the ability to read each other's minds telepathically, you and I.
Yes, connected.
I would get a bunch of words that I could read. You
Not
would just
images.
get a bunch of pictures. You would get a whole bunch of images that you would have to then make some sense out of why they are there.
But she could, because it would be your brain, so it'd be fine.
Well, no, I mean, but if I'm saying I had to swap that because that wouldn't.
And then I'd
would
be
just
like,
the
what's
other
that
person.
weird feeling in my left shoulder? And it would, it would
Why is
actually
it
be that
blue?
his
Why
body,
is it
his
green?
nose is itching, but my body
Yeah, so no, we
his
can't
brain are
swap
not
brains
connected the
because then we
same.
would just have a different head on the. But no, if we would telepathically reading each other's thoughts, I think I'd have a better shot of understanding because I'd have a bunch of word association that I could play with because you think in words. But you would have a tougher job, I think,
Thinking images.
because you would get a lot of like, where is this hallway? What is this? I have all these. I have spatial places and I have physical objects. I have pictures of things. I don't have words, which is why I've always like been terrible speller. Can never remember a name. I can't see it. I don't see the words.
In this one case, you could read me like an open book.
But if I could, your mind, I could read like a word cloud.
Like a word.
Mine would be a collage of imagery.
I wonder if
beautiful pictures.
If like brain processing would be so much slower because you'd have to like
work it out.
Yeah.
What,
even though you'd be reading something, your brain is not accustomed to functioning and thinking things up by reading, so it would still take longer than whatever your brain is used to doing.
Because I think if I can picture the word, I think that's what happens when I read like a book. So I think it would be quick. But it's very curious because you know how people say in a dream you can't read things? I have a hard time thinking of a word, seeing a word, just visualizing a word. It does the same thing it does in the dreams. It's like it's assembled and now it's gone.
Yeah, that's also complete nonsense because I read in my dreams all the time. So
no, I can't. And I have things that will come up like, oh, here's a map to where we're going. Okay, I'll just directions to where we're going. And oh gosh, Oh yeah, that's right. I'm in a dream. I can't read any of this because it's a dream. What do I take and looking at anything written down for? So but now Blair, you're a little bit of a hybrid because you say you think in pictures,
I do.
but you can read in dreams.
I can read in my dreams. It's no problem at all.
Wow! So you can visualize words.
Mm-hmm.
I cannot in any form, which is why I could do like in primary school when they start testing you, they have like, I don't know, what is it, 8th grade, 7th grade test, or there's like a big test in California somewhere where they test every kid. I got 99th percentile in English reading comprehension, like almost a perfect score. Almost just knocked it completely out of the park. I think I missed like one question. My spelling was something atrocious, like 60, 54, 55%, something like that. It was like remedial, as though I could not read. And it's just, I have no memory for if that word looks close enough, I'm good. I'll just roll with it.
It's good enough. It's phonics, right? It's close enough.
That's good. Partially because English is stupid.
Right. So, well, this particular study, though,
that I can't proofread. I also can't proofread my writing because of this, because I can't tell the difference. Oh God!
Proofreading your own writing's hard anyway. You need somebody else to do that. Okay. But this particular study, though, they're trying to suggest that by bringing in this idea of relativistic phenomena in physics and developing a mathematical foundation, a formalization for consciousness that hopefully bridges this gap in the hard problem of consciousness, figuring out where it is, that maybe in the future, philosophers can usefully contribute to the science of consciousness in collaborations with neuroscientists to study the neural basis of these phenomenal structures. So potentially, maybe philosophers will finally be able to work with neuroscientists and come together through the common language of physics, which, if that can happen, that itself is solving a hard problem in the world of science.
I just feel like you don't need the hard science. I know everyone wants to attach cosmetic products to quantum physics science. Stop.
No, people want to know what's conscious. Is a rock conscious? I don't know. What is consciousness? Is it quantum? I don't know. People want to, scientists ask the questions. Whether or not we have the tools currently or the understanding, we're working on it. Slowly chipping away.
Philosophers want to be a hard science, but there's not there's no
philosophy
has a place. It's not hard science.
Yeah. Anyway,
it led to a lot of hard science.
I think we've made it to the end of the show. We've made it. We made it to the end of the show.
Stop science shaming, Justin. Let's close out the show.
Let's close it out. Philosophy is a science. It is.
Attacking social science last week, philosophy this week. What are you up to?
No science shaming. No. We've come to the end of the show. Yeah.
Is considered science?
It's time. Are we done? We're done. Are we done now? We're done. Yes. It's time. Yeah, hey, everybody. Thank you for joining us for another episode. We are so glad that you were here with us for the whole thing. I know you stayed for the whole thing, and I hope you enjoyed the show. I want to give shout outs to Fada and for his help with social media and show notes over on YouTube. Identity 4, thank you for recording the show. Gord, Arnlor, others, thank you so much for keeping it nice, compassionate in the chat room, keeping it real over there. Rachel, thank you so much for your editing and the work that you do. And thank you so much for our Patreon sponsors and their help in what they do in allowing us to do this show. Thank you to Teresa Smith, James Schaefer, Richard Badge, Kent Northcote, Rick Loveman, Pierre Velazar, Ralphie Figueroa, John Ratnaswamy, Carl Kornfeld, Karen Tazi, Woody MS, Chris Wozniak, Dave Ben, Vigard, Chef Stad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Garv Sharma, Reagan, Derek Schmidt, Don Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredus 104, Sky Luke, Paul Ronovich, Kedden Ridden, Noodles, Jack, Brian Carrington, Matt Beto for Texas, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Ramide, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Philip Shane, Kurt Larson, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, Tony Steele, and Jason Roberts. Thank you. Thank you, everyone, for your support of TWIS on Patreon. And if you are interested in supporting TWIS on Patreon, head to twis.org and click on that Patreon link. On next week's show. Actually, on tomorrow's show.
Oh, huh?
Yeah.
We have a show tomorrow? What day?
I have an interview tomorrow, 11 a.m. Pacific time. I will be speaking with Dr. Moya McTeer, and she has a book out about the Milky Way. I am very excited to be speaking with Dr. McTeer about her work.
I heard that about that. That's the galaxy that's really, really close to us.
Yeah, it's a close one. Everyone should have something that they find in common with that. But aside from tomorrow, Thursday, August 18th,
we will
at 11 a.m. Pacific time.
I'll be back here together,
Yes.
8 p.m. Pacific time, broadcasting live from our YouTube and Facebook channels, as well as twist.org/live.
Hey, want to listen to us as a podcast? Maybe as you walk around and take pictures of orb weaver spiders up to no good? Just search for This Week in Science where our podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to the stories, so you can read them yourself, will be available on our website, www.twist.org. And you can also sign up for our newsletter.
You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, into the subject line, or your email will grow its own consciousness and go away.
To Twitter, where we are at Twist Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We'll love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.